Okay, this video is going to go over the expectations for the second of the IA criteria. These are for first exams in 2025, and this criterion is called data analysis. Okay, so there's basically three strands that we're going to look at when we look at the data analysis and when you're assessed for your data analysis score. The top score is 6 points. So if we look down here, we can sort of see the three strands listed here. So the communication of the recording and processing of the data is both clear and precise. The recording and processing of data shows evidence of an appropriate consideration of uncertainties, and the processing of data relevant to addressing the research question is carried out appropriately and accurately. Okay, a few more notes that they've given us for clarification. Data refers to quantitative data or a combination of both quantitative and qualitative data, meaning observations. Clear communication means that the method of processing can be understood easily. Precise communication refers to following conventions correctly, such as those relating to annotation of graphs, tables, use of units, decimal places, and significant figures. Consideration of uncertainties is subject-specific, and further guidance is given in chemistry teacher support material. So those are uncertainty rules that we've gone over in class. Major omissions, inaccuracies, or inconsistencies impede the possibility of drawing a valid conclusion that addresses the research question. The conclusion is in the next criterion, but they're just mentioning it now, that obviously if you leave things out or you make major mistakes, it'll be really hard to draw a valid conclusion. Significant omissions, inaccuracies, or inconsistencies allow the possibility of drawing a conclusion that addresses the research question, but with some limit to its validity or detail. So I guess they're saying major is worse than significant, but either way, if you're leaving things out or making mistakes, it's going to make it hard to do a good job in the next section of the lab. Okay, so let's break those back down in a little bit more simple language. I would think of your data analysis section as sort of having these components. So you're generally going to start it with your raw data and your observations. The key here is having clear, organized data tables that are really well labeled with units, so it's really clear what all the data is. It's also really important that you don't leave out what's called the raw data. So sometimes when your data is kind of simple, or some of the data is simple, like let's say you have a initial volume and a final volume that you're measuring. Sometimes students think it's why even bother reporting those? Let's just go straight to the change in volume. But it's really important that you include the initial raw data as well, the actual values that you measured, volume initial, volume final. This also includes things like if you use a data logger, if you're using Pasco and Capstone, include a sample screenshot. You don't have to include all of your Pasco graphs. You might have 15, 25. That would take up your whole, your whole IA, so you don't have to do that. But a sample screenshot of data logger data is also considered evidence of raw data. And then observations, those are the qualitative bits of data. I would suggest always including qualitative observations of some kind. The more specific they are to particular treatments and trials within your lab, the better. But just having that section there as a general practice, because you can draw on those observations later in your conclusion. They can help inform method evaluation, and they can help inform what you're able to conclude. They can support that. Okay, so first we're going to show our data that we've collected, and we're going to show our observations. Then we're going to move on to processing that data. That means you're going to present your calculations and you're going to explain what you're doing. It could be as simple as presenting a formula, right? Like let's say we're using the ideal gas law. So you write PV equals nRT, and then you're going to show plugging in values after that. Depending on the processing you're doing, you might need to justify or explain a decision that you're making. So it's always good to explain your thinking. But if it's sort of a obvious everyday chemistry approach, like converting from grams to moles, you don't really need to explain your thinking. You can just show the processing that you're going to do. Make sure it's clearly presented. Make sure it includes units. ISA within the calculations will help the clarity of the presentation. And as mentioned above, they want you to watch out for rounding rules and significant figures and things like that throughout. So just really careful presentation of your working out, right? This will be your one big lab report that you submit to the IB. And so you'll want to really take the time to make sure you're following all those rules carefully and that you're presenting things really clearly. And then you have to also present uncertainty processing. So the expectation is you will be considering uncertainty in your investigation and its impact on what you can conclude. And in order to do that properly, you need to show how you are processing uncertainty through your results. This may be as simple as you've decided to do the half the range method on all your trials. You may be doing the method from class where you're converting all of your measurement uncertainties into percentages and combining them and then converting them back to absolute uncertainties. You're going to give a little bit of explanation again of your thinking behind your method, and then you're going to show a sample calculation. And that goes for strand 2 as well. We're talking about sample calculations. We're not doing every single trial, every single treatment. If we are doing certain calculations, it's important that we show each type of calculation once, where we explain what we're doing and we plug in values, but then you don't have to show every single calculation. Things that aren't really mentioned in that criteria above, but which are expected, is that after you've done all your processing, you're going to put all your processed values into a data table to summarize all the work that you've done. So if you've got 15 or 25 total trials, you're only going to show one calculation, but all 15 or 25 should end up, the final values and the final uncertainties should show up being presented. And so a data table makes the most sense. And most IAs will end with some kind of graph. In chemistry, they tend to take the form of scatter graphs, right? Maybe we're looking for a linear relationship. Maybe we're looking for a negative linear relationship. Maybe we're looking for an inversely proportional relationship. Maybe we're expecting an exponential relationship of some kind. It depends on your topic, but usually you're going to have a graph with your independent variable and your dependent variable, and everything should be labeled. There should be units. We should include equations that we get for these trend lines. We'll have error bars representing our uncertainty. That's kind of the final, I consider it like the visual answer to your research question. In most cases, your IA will be, how does a particular independent variable affect the dependent variable? And the graph gives a nice visual representation of that answer, and also gives a sense of confidence based on things like the fit of the line for the points, and the error bars representing how much precision, how much uncertainty you have. So quick to look at a couple of exemplars here. These are all labs. They are from the former syllabus, so these are not on the exam 2025 technically, but some of these were moderated by the IB, and all of these achieved 7s, and they had a 6 out of 6 in this analysis or data analysis criterion. So you can see this student has included a screenshot from their data logging. They've shown how they did processing on that screenshot, and they've included a title and a description so I know what I'm looking at. Then we've got a raw data table here. You can see very clear labels. We've got units, and we've got uncertainties. Okay, they've been careful about precision here. All the values have the correct and the same precision within a given value. So it's just a nice data table. It's got a title that labels everything that's in it. Then we move on to observations. This student chose to do processing in a table. So on the left-hand side, they put the formula that they were going to use. And then on the right-hand side, they plugged in actual values. And notice it says sample calculations for treatment 1. So they're even telling us, okay, I'm using the values from this particular treatment as my sample calculations. To end, they've got a process data table that sort of summarizes all of their processed values. And then in this particular lab, there were 2 different graphs. I haven't shown the full lab. There are 2 different graphs here. You can see the graphs have clear titles. The axes are labeled. They've got these scatter graphs with trend lines. The trend line equations are included down here. They've included these R-squared values, which we'll talk about further, but these are ways of considering how good the fit is between your line and the data points. And we've got these error bars representing the uncertainties. Okay, so a fully presented, clearly presented, really nice data analysis section. Just to show you quick some other examples. So this is a different full data table, right? Again, really clearly labeled, consistent precision, uncertainties, units, labels, observations that included a picture for this one. Then just a different way to do calculations. They don't have to be in a table, but similarly, we've got an explanation of what formula we're using and then an example calculation with specific information about what values are being plugged in and so on. Calculating uncertainty, the method is explained to an extent. I mentioned here they could explain the method a bit more, their reasoning here a bit more, but they've shown a sample calculation. They have a process data table and they have final graphs. Okay, and one final one, again, a sample calculation or sample graph from data logging with clear labels, a raw data table. We've got observations. We've got some processing. Again, this is not in the table format, but each step gets a little explanation, and then we're plugging into an equation. Then we get an explanation, and then we're showing the equation here, and then plugging in right next to it. That's another way that it would be nice and clear. And then this person has used a different program than Excel to do their scatter graph, but they've got their data points along the way with their error bars right here. So they're doing that. They've actually added max-min trend lines, which is sort of a physics expectation and is something that you can strive for in chemistry if you want to look at the uncertainty on your trend line slope. But either way, those are three really good examples, 6 out of 6 data analysis sections. So pick one that works for you and try to emulate the level of clarity, the different sections, and you'll be in good shape for your data analysis.